Hello, everyone. This video will be a presentation about a recent research paper that was published in Nature focusing on a new FDA-approved drug for Alzheimer's disease. First, I want to give a brief background on Alzheimer's disease. As some of you may know, it is a progressive neurodegenerative disease, meaning a disease that gets worse over time that impacts the cells of the brain, thereby impacting memory. This disease affects about 1 in 9 people over the age of 65, and it is hard to diagnose. Marks of Alzheimer's disease is the buildup of something called amyloid beta plaques. Amyloid beta is a normal protein found in the brain. However, in Alzheimer's disease, or AD, amyloid plaques form, which disrupts normal cell function. This is thought to be one of the main causes for memory loss. As you can see here in this image, the red dots are the amyloid beta plaques. Mentioned before, the FDA recently approved a drug for Alzheimer's disease. This drug is called aducanum. However, the drug has been called into question in the scientific community in terms of the lack of effectiveness in combating the symptoms of Alzheimer's disease. Research, along with some harsh side effects of the drug. There's ongoing research being done to try and improve the outcome of the drug, which is what this recent research paper, Meningeal lymphatics affect microglial responses in anti-amyloid beta immunotherapy by Mesquita et al. Meningeal lymphatics are essentially a drainage system for the brain, helping to keep a balance of fluids in equilibrium, and also helping to keep the immune system optimally working. Microglial cells are the body's natural immune cells. And anti-amyloid beta immunotherapy essentially just means a drug that is trying to get rid of amyloid beta. On to the main methodology of this paper. The researchers used an animal model system in which all mice had Alzheimer's-like conditions and were receiving weekly injections into the brain. The difference within the group of mice are what they were being injected with. Group 1, also known as a control group, were mice that were injected with a saline solution. Group 2 were mice that were injected with the mouse version of the human Alzheimer's disease drug. So this is the mouse version of the aducanum. Group 3 were the mice injected with and under conditions that negatively impacted their meningeal lymphatic system, or they just had damaged lymphatics. Group 4 were mice that were injected with and under conditions that negatively impacted their meningeal lymphatic system, but they were also injected with the mouse version of the human Alzheimer's disease drug. So there is a relationship between groups 2 and groups 4. So why does one of the group of mice have a damaged meningeal lymphatic system? It is thought that the impacted system may make Alzheimer's disease progress faster or have a more severe decline. So studying the effects of the dysfunctional meningeal lymphatic system may help to collect more information about how this impacts Alzheimer's disease, and also potentially may give some reasons as to the effectiveness of this drug. Moving on to one of the figures of the paper. So these were groups 1 and group 2, as mentioned before, and then also included were groups 3 and group 4. So just a reminder, group 1 was the one who had no Alzheimer's disease drug. Group 2 was the one that had the Alzheimer's disease drug injected. Group 3 was the one who had no Alzheimer's disease drug but had the damaged lymphatic system. And group 4 was the one who had Alzheimer's disease drug as well as damaged lymphatic system. So these images were taken through a fluorescence microscope of a slice of a mouse brain. Every red dot that you see is an amyloid beta plaque. Remember, this is something that makes the neurons in the brain not very happy. Group 1, you can see some red dots. There are some amyloid beta plaques, but there are not too many. This is also Alzheimer's disease, so they are naturally occurring within the disease, so this is expected. Group 2, you can see that there are some red dots, but they are significantly less red dots than group 1. Moving on to group 3, there are many red dots. So there are many amyloid beta plaques. This is within the damaged lymphatic system. So showing that with the damaged lymphatic system, there are an increased amount of amyloid beta plaques as compared to group 1. Moving on to group 4, there are less amyloid beta plaques than group 3. However, if comparing group 2 to group 4, which both have Alzheimer's disease and both have the Alzheimer's disease drug, you can see that there are more amyloid beta plaques within the damaged lymphatic system. Moving on to this figure, these are the same categories as before, but they are looking at the overall neuronal health. So if there are more green dots, the neurons are more stressed. Overall, comparing this group over here to this group over here, overall comparing the groups, it seems that within the damaged lymphatic system, the neuron seems to be more stressed. So what is this paper saying? The data in this paper demonstrates that the damaged lymphatic system causes overall stress to the brain. Therefore, they can cause an increase in other areas, such as amyloid beta and overall neuronal health that was decreased. Finding a way to therapeutically alleviate the stress or symptoms of the damaged lymphatic system could lead to this drug having potential better clinical outcomes overall. Thank you so much for watching, and I hope that you learned something new.